Hello everyone and welcome to Lab Manager's Tech Trends webinar series. My name is Trevor Henderson, Technology Editor for Lab Manager, and I will be moderating today's discussion, which will focus on managing sample prep for chromatography. Today we have a panel consisting of 3 leading industry specialists who will share their perspectives on some of the new tools and protocols to minimize and simplify sample prep and how to effectively choose which methods work best for your analyte, your separation requirements, and your instrumentation. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the webinar, and the panelists will address these during the question and answer session following the presentations. To ask questions, simply type your query into the question box located at the bottom left-hand side of your screen. We'll try to address as many of these questions as possible during the Q&A session. However, if we happen to run out of time, I will forward any unanswered questions to the panelists and they can respond to you directly. Additional resources can be accessed through the resource widget located at the bottom of your screen. You may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. You may need to move or minimize some of the open windows to see the live view. This webinar recording will be available early next week on Lab Manager's website. At the end of this webinar, we'll share that link with you, as well as contact information for all of the panelists. With that, let me introduce our first speaker. Joan Stevens earned a Bachelor of Arts in Chemistry at the College of Worcester, a PhD in Medicinal Chemistry from the Ohio State University College of Pharmacy, and conducted postdoctoral research at Purdue University. She joined Agilent as a sample preparation application scientist in the Chemistries and Supplies Division in 2009. And prior to Agilent was with Gilson, Inc., where she worked from 1989 to 2009 in technical sales and support as a technical support manager and applications manager. During her 25-plus years, Joni worked closely with customers, sales, and technical support personnel, as well as state, federal, and local government agencies on various projects. She was directly responsible for work and applications that were presented in technical conferences in the U.S., Canada, and Europe. Multiple applications were also published in the areas of sample preparation for food, pharma, bioanalysis, and chromatographic method development. Since being at Agilent, Joni has been an integral part in Agilent's success in becoming a market leader in catchers. She has also produced applications in bioanalysis, pharma, and food. She has published over 35 application notes since 2009 and has been directly involved in 27-plus application collaborations and presented work at all major conferences. Joni's goals are to involve researchers actively in sample preparation and therefore eliciting an understanding of how sample preparation can optimize research and results. Joni, thanks for joining us today.
Thank you, Trevor. And thank everyone. I really appreciate you joining us today. My topic, my first talk, is going to be on sample prep toolkit, choosing the correct technique. Today, we're going to have an overview of sample preparation options, discuss how to select the right product, and then our take-home message regarding sample prep. So let's begin with our sample prep toolkit. Without proper sample preparation, you may damage your instruments, which can be costly. For example, increased instrument downtime, maintenance and repairs. Parts like UHPLC columns, MS capillary, quadrupole pulp seals, injection ports, the list continues. And also, in addition, service charges. Without sample preparation, there is a high chance of contamination and carryover issues. As shown in the figure, without proper sample preparation, you may not see the peaks clearly, or even if you see them, you can't quantify them for your analytes in the sample. In the latest version of Analytical Scientists, a group of sample prep scientists were discussing sample prep in general. And Frank David from the RIC stated in the article, "It is clear that errors made in sampling, sample preparation, or sample introduction, like injection, cannot be corrected by using even the most advanced MS system." This drives home the point that sample preparation is very important. With that being said, our goal is to try to strike a balance between effort and investment and quality of results. This is sample preparation option decision tree based on application needs. For application needs, ask yourself, do I need absolute trace or residue level of analysis. If residue level detection is what you need, you don't have to make your sample ultra clean. So catchers, SLE, filtration, including Captiva and Lipids, will be appropriate starting point for your sample preparation needs. If absolute trace level is needed, SPE is going to be your first choice, and in some cases, SLE will also work. When we look at the decision tree based on instrument type, we can think about our sample prep from two perspectives. With tandem MS instruments such as triple quads or ion traps, additional selectivity helps discriminate between target compounds and the sample matrix that can co-extract. This means that the sample prep method that you choose could be simpler and do less sample cleanup. Essentially, the mass spec is taking care of the role of eliminating the matrix. Sample prep methods that are less selective, like catchers, dilute and shoot, or SLE, can be great choices to match up with tandem mass spec. Of course, the sample matrix is still there. Only the MS-MS experiment makes it look like it's not. So if you find that your instrument performance is declining, LC column back pressures are increasing, chromatography shifts, response is dropping, a more rigorous sample prep method such as SPE might provide added sample cleanliness. For less selective detectors, like single quadrupole mass specs or conventional chromatography like UV diode array electron capture detectors, using a more selective sample preparation technique such as SPE can give additional power to identifying target compounds. So in general, as we move down this list from direct inject, dilute and shoot, filtration, onto SLE, which is solid supported liquid extraction, catchers, solid phase extraction, and immunoaffinity columns, you are going from less selective to more selective type sample prep. This is only a subset of the available sample prep techniques, but today what we're going to do is focus on the following. For instance, we'll start with filtration, which is removing basic particulates from all types of samples, but when you mix that with a functional filtration, you can remove lipids. Solid supported liquid extraction increased productivity relative to liquid-liquid extraction, and it can be automated, and it's ideal for aqueous samples. Then we'll look at catchers, which is an easy-to-use sample preparation for food testing. It can be solid, liquid samples, water-rich, water-not, like vegetables, fruits, meats, seafoods, and solids. And then we'll end with solid phase extraction, which will offer ultra-clean sample preparation for the analysis when high selectivity and sensitivity are required, such as for trace level analysis to protect your highly sensitive instrumentation. Okay, so what are we talking about when we mention filtration options? Well, for instance, the main point about filtration is to retain particulates from your sample prior to injection. Agilent has different filter materials, pore size, and dimensions to meet your needs and requirements. ND, that you see in the middle of the screen, stands for non-drip, which means until vacuum positive pressure or centrifugation is applied, the filtrate will remain in either the well or the cartridge. So therefore, you can do protein precipitation directly in the well before applying vacuum positive pressure or centrifugation. In the case of Captiva and D-lipid, this incorporates the mechanical filtration that we talked about, but has a built-in lipid removing sorbent that will retain lipids from your sample. Well, why filter? Well, first of all, you can remove particulates prior to solid phase extraction. That's especially important with really terrible water samples. Particulates can be removed prior to dilute and shoot or direct injection. And on reconstitution, in the mobile phase, many times you will get precipitate particles falling out of solution. So you want to remove those before you inject into your system. In this slide, these are the steps associated with the Captiva ND lipid. First, you're going to add your crash solvent, usually a 3-to-1 ratio to sample, and then add your sample. Mix with pipette or automated liquid handlers. The 0.22 micron filter will retain particulates formed from protein precipitation, and the lipid stripping sorbent will remove lipids. The sample will not pass through the unique filter material until vacuum, positive pressure, or centrifugation is applied. Now the sample is actually ready for analysis. This is an example of the benefits of using both functional and mechanical filtration, as I mentioned in Captiva ND lipid. The top trace results from post-column infusion of albuterol. The area circled in the chromatogram shows ion suppression of the albuterol signal. Negative peaks relative to where the phospholipids elute on the bottom trace is where you're going to see ion suppression. In the lower chromatogram, after using Captiva ND lipid, we see substantial improvements in the albuterol signal since ion suppression is nearly absent. We need to keep in mind that protein precipitation does not remove lipids, whereas the Captiva ND lipid removes particulates, proteins, and lipids. So let's compare the time savings. The time required to prepare 96 samples by a Captiva ND lipid plate is 5 minutes, whereas with proteins involving protein precipitation and centrifugation, the time is 26 minutes, and there is still lipids in the sample that will build up in your analytical system over time and cause chromatographic issues. Here's just a simple example of using Captiva ND lipid for sulfonamide residues in milk. We're showing excellent accuracy and RSDs for the range evaluated, and look how simple the procedure is. Now let's take a brief look at solid-supported liquid extraction. The mechanism is very similar to liquid-liquid extraction, which all of us are familiar with. However, we use much less solvent, no emulsions will form, so it takes much less time and improved data quality, minimal method development, and it's automation friendly. The basic procedure is to pre-treat the sample if it's not in an aqueous form, add the aqueous sample to an SLE cartridge, no conditioning is required, wait until that aqueous sample absorbs. Now at this point, that takes 5 to 10 minutes, so you can check your email, have a cup of coffee, chat with your lab mates, and then come back and elute with a water-immiscible solvent, such as DCM, MTBE. This is actually the liquid-liquid extraction step that you're familiar with. And then elute, when you elute, this collection is ready for GC injection, or if you're going to LC, you will need to evaporate and reconstitute in the appropriate solvent. Here's an example of using SLE for pesticides in honey. Specially highlighted in the blue circles are some compounds which are not even detected or extremely low by conventional liquid-liquid extraction, highlighted by the red bars. The SLE are the yellow bars. So we see that SLE gives us great recovery in most cases relative to liquid-liquid extraction. Other examples are azo dyes in textiles, caffeine in black tea, polyphenols in wine, phthalates and whiteners in cosmetics. Now let's move on to catchers. Catchers is quick, easy, cheap, effective, rugged, safe sample prep. It is one of the hottest trends in sample preparation, especially for food testing, import-export, food additives, contaminants, residues, colorings, and all types of food and food-type products, and now is even being moved on to other areas. The list is endless, and it's currently expanding to other application areas daily. Depending on your matrix and analytes, catcher salts and dispersive SPE are available in pre-packed packages with appropriate extraction materials and sorbent. So the bottom line, if you can pipette and you can shake, you're able to analyze your samples by catchers. For complicated food matrices or special food applications, catchers can also come in a customizable packaging. Initially, what we see in this top chromatogram is what a lab was doing to analyze their lemon oil sample. They diluted it 1-to-1 with hexane. The matrix components are clearly seen, and finding target compounds would be challenging. By performing a simple catcher's extraction and cleanup procedure, the sample matrix is removed and target compounds are readily identified. The top trace shows lemon oil sample that was spiked with pesticide mix at 200 nanograms per mil prior to extraction, while the lower trace shows a lemon oil sample that was spiked with the same concentration of components after extraction. Highly clean sample. Other examples are pesticides in tea, acrylamides in fried food, PAHs in seafood, PCBs in fish, mycotoxins in grains, antibiotics in liver, vet drugs in pet food, edible food, and also components in liver, pesticides in rice, pharmaceuticals in whole blood. Now, here's an example of what I like to call a 1-2 punch approach. This is using the catcher's procedure, but after the dispersive, pass the sample through a Captiva ND lipid to remove additional lipids that might be in the sample. Not only does this remove lipids, but at the same time mechanically filtering the sample prior to analysis. And this can be done directly into the autosampler vials. This avoids particulates from the dispersive inadvertently being transferred to the autosampler vials and removes additional lipids. This approach improves recovery as shown by the data for pesticides from the matrices of fish, pork, milk, and egg. You see that it reduced ion enhancement and ion suppression as shown here by making the values 80 to 100 and 72 to 117% in the acceptable range. And this is by GC-MSMS. Data from an LC-MSMS for antibiotics in fish, poultry, milk, and egg also shows that we reduce ion suppression to the acceptable range. So it's very nice to use this 1-2 punch approach for your analysis. Now let's move on to our last area, which is solid phase extraction. There are many types of solid phase extraction. For instance, reverse phase, cation exchange, which works really well for basic compounds, anion exchange for acidic compounds. Mixed mode is always my choice if I can because it combines reverse phase and ion exchange. And there's always specialty sorbents like carbon, an SPE designed for mycotoxins, or atrazine. Solid phase extractions has a lot of capabilities. It's very selective and sample prep technique. Highly clean samples are achieved, wide range of applicability from food, environmental, to pharma, and it is automatable. For this example, this is an anion exchange column, bond-dilute SAX, was used to prepare drinking water for the analysis of haloacetic acids. Two different GC columns were used to compare resolution and suitability for the method. The results shown here is a fortified reagent blank at 10 nanograms per mil. The bond-dilute SAX even gave excellent recoveries for the trihalo compounds, which are known to be problematic and has been pointed out in the literature. So why would you want to choose SPE? Well, first of all, it's flexible. It can match a broad spectrum of sample and target compounds with different sorbents and forms. It has a wide range of formats and sorbents for low detection limits and longer instrument lifetime, and cleaner extracts. Increased sample throughput with the automatable friendly formats, and it's really easy to adopt because there are so many publications and applications that are available that you can use as references. One idea is to actually take SPE and make it online. What's the advantage of this? By using online SPE allows you to use a much smaller sample volume, for instance, water, and all of that volume can be prepared at once. Combining this technique with sensitive MSMS offers extremely low levels of detection for emerging contaminants. The logistics of collecting the samples, for instance, water, which used to be hundreds of mils of volume, are now drastically simplified by the online approach. Here's an example of how this is done, and this is with the Agilent Flex Cube. If you'll notice here with the red flow path, At the beginning of the analysis, the piston is delivering the sample from the autosampler to the trapping column, SPE1, and that is identified by the red flow path. We switch the valve, so after loading the SPE1, the right valve is switched and the SPE1 is moved in front of the analytical column, now becoming the blue flow path. The SPE2 is cleaned and equilibrated for the following sample, shown by the red flow path. In this slide, we see excellent peak shape for cyanidin for the calibration range shown, 1 to 50 nanograms per liter. On the right, we see that we are able to use this method to actually determine cyanidin in collected samples from regional tributaries. The table here shows that for the 20 pesticides evaluated, we have excellent recovery and RSDs, all below 7%. Other Agilent sample preparation options that we have are GPC cleanup, headspace, purge and trap, SPME, SPME, and flash. So therefore, with the numerous sample preparation techniques that we have quickly presented to you here, it is possible to strike a balance between effort and investment and quality of results based on your application requirements with the correct sample preparation technique. This slide shows you a few references that I would like to suggest that you either download or request, especially the first one, which is a sample preparation handbook written by Ron Majors. It is over 300 pages of sample preparation techniques with multiple references. This is a must-have in anyone's lab. And please feel free to contact me directly if you would like. My email is noted below, and all of these will be found on the resource area of this seminar after it's been published. And with that, I'd like to thank you and wish all of you happy sample prepping.
Thank you very much, Joni, and thank you all for sending in your questions. I encourage you to continue to do this throughout the presentations. If you're joining us late, you may ask questions by simply typing your query into the question box, which is located at the bottom left-hand side of your screen. Our next speaker is Jennifer Claus, who joins us from Supelco. After receiving a Bachelor of Science in Chemistry and Biology from Lycoming College in 2001, Jennifer began her chemistry career in the medicinal chemistry department at Merck & Co., where she synthesized potential drug candidates for projects targeting type 2 diabetes, anemia, and obesity. While working, Jennifer continued her education to receive a Master's of Science in Chemistry from Rutgers University. Jennifer joined the Analytical Research and Services Group at Supelco in 2007. Her role as a chiral application chemist focused on providing comprehensive laboratory-based chiral services, including chiral screening, method development, and preparatory chiral separations to customers in support of Aztec and Chromasil product lines. In 2012, Jennifer joined the marketing group at Supelco in the solid phase extraction product manager, becoming responsible for the worldwide marketing of SPE products and accessories. Thank you for joining us today, Jennifer.
Thank you, Trevor, for that kind introduction. As Trevor mentioned, my name is Jennifer Claus. I am the SPE product manager here at Supelco Sigma-Aldrich, and we're going to talk about a very difficult sample matrix, edible oils. We'll talk about the sample cleanup for the analysis of compounds in edible oils. More specifically, we'll go over some of our current methods for oil cleanup prior to POP, or persistent organic pollutant analysis. Specifically, we're going to talk about PAHs, or polycyclic aromatic hydrocarbons. Then we'll get into a new SPE approach for this cleanup. We'll talk about how it works and give an example application. So in a recent survey to food analysis chemists, we asked, what type of sample matrices proved to be the most challenging? We found that 59% of the respondents indicated that fatty samples or oils were among the most challenging. The analysis of oily or fatty samples presents a challenge due to the heavy matrix effects. In the case of GCMS, fatty matrix can cause contamination of the GC inlet, column, and detector. So what's the issue? Why should we analyze for PAHs in edible oils? Olive oil, for example, can become contaminated with polynuclear aromatic hydrocarbons, or PAHs, through exposure of the olives to pollution in the environment. Concern over exposure to these compounds has caused some countries within the European Union to set limits on PAH content in olive oil. In 2011, the European Union Commission Regulation number 835/2011 set a maximum limit in various foodstuffs, including edible oils. The limits for oils and fats, excluding cocoa butter and coconut oil, have been set to 2 nanograms per gram for benzo-A-pyrene individually and 10 nanograms per gram for benzo-A-pyrene, benzo-B-fluoranthene, crystine, and benzo-A-anthracene combined. Now, a number of methods currently exist to analyze PAHs in edible oils. For example, there are some ISO methods, 15302 and 15753 for benzo-A-pyrene only. This uses alumina column for extraction, and 15753 uses 16 PAHs from light to heavy using liquid-liquid extraction followed by a 2-step cleanup with C18 and Fluorocell. Other methods include liquid-liquid extraction followed by gel permeation chromatography, or GPC, silica gel or Fluorocell SPE using large glass columns, or molecularly imprinted polymers, or MIP SPE. These other cleanup techniques sometimes can be time-consuming, expensive, or provide inadequate cleanup. As far as the market needs, POP analysis in edible oils, the cleanup must be suitable for a full range of PAHs from 2 to 6 rings, have good overall performance, and this includes sample cleanup selectivity, analyte recovery and reproducibility, method simplicity, amenability to GCMS or HPLC fluorescence, and method robustness. Recently, a new approach has been developed for the analysis of nonpolar POPs in edible oils. This includes the use of a dual-layer SPE cartridge containing Fluorocell as a top layer and ZSEP C18 as a bottom layer. The Fluorocell layer retains polar compounds such as fatty acids. The ZSEP C18 layer on the bottom retains the fatty matrix through both Lewis acid-Lewis base interactions as well as hydrophobic interactions. As you can see in the picture on the left, you'll notice a yellow color on the cartridge. This is oil matrix removed from the sample. So basically, the unwanted oil matrix is retained on the cartridge while the analytes of interest are eluted. The final extracts are compatible with GC or HPLC because they're all in acetonitrile. So let's get more into ZSEP C18. What is it? Well, ZSEP is basically silica with zirconia bound to it. C18, we all know, is C18 on silica. If you mix the two, you have ZSEP C18. Now, the retention mechanism, we believe, is as follows. First of all, the C18 retains fats through hydrophobic interactions of the carbon chains, and ZSEP retains the fats via Lewis acid-Lewis base interaction with the zirconia and the hydroxyl groups of the glycerols. Now let's take a look at an example application, the extraction of PAHs from olive oil. Essentially, this procedure is only 3 steps. First, you condition the cartridge with acetone and let it dry. Add the oil and internal standard to the cartridge, and then pull acetonitrile through the cartridge and collect the eluent. You can then adjust the volume via addition or evaporation of the acetonitrile and proceed with HPLC fluorescence or GCMS analysis. Here we have an example of a chromatogram in selected ion mode done by GCMS for the analysis of olive oil. Now, this was spiked with 17 different PAHs. As you can see, due to the absence of background, we were able to quantify and identify the 17 PAHs. Note that this is done on a single quadrupole MS. at a higher mass spec temperature to minimize the tailing of the heavy PAHs, and a pulse injection was done to increase the response of the heavy PAHs. As far as recoveries, you'll note that the recoveries are greater than 80% for the most of the PAHs. The relative standard deviations are less than 15% for most PAHs, and this method was acceptable for the detection at 2 nanograms per gram on a single quadrupole MS instrument. A method ruggedness test was done to test the cleanliness of the extract. After spiking the sample with 28 different PAHs having a variation between 2 to 6 rings and making 110 injections of olive oil onto the GCMS system, it was found that some loss of response was seen, but 17 of the 28 PAHs decreased in response by less than 20%, and all the PAHs were still easily detected at the end of the sequence. So, this next experiment compares EasyPop NP to other SPE methods, including silica gel SPE and MIP SPE. The experimental details are outlined below. Comparing the GCMS background, you'll notice that EasyPop NP has much less background than silica gel SPE and MIP SPE. In fact, the background remaining in the extracts prepared using silica gel SPE and Supelco makes them unsuitable for a rugged GCMS analysis. This bar chart compares PAH recoveries between EasyPop NP, MIP SPE, and silica gel SPE. Compared to silica gel SPE, the EasyPop NP gives better recoveries and overall lower background. Supelco will produce better recoveries for the heavier PAHs between 5 and 6 rings than EasyPop NP, but more background can be expected for lighter PAHs. For lighter PAHs, the heavy background prevents quantitation in most cases. Notice that all the pH recoveries for the smaller PAHs for the MIP SPE kind of look like they're off the charts. However, this is because of matrix effects. You'll note that there's a lot of co-eluting matrix interferences for the Supelco cleanup for 2 to 3 rings. High recovery values are inaccurate due to the matrix effects. So in conclusion, the new SPE method using the EasyPop NP is suitable for the extraction of a full range of PAHs between 2 to 6 rings, the detection of PAHs at levels specified in European Regulation 835/2011. It produces an extract that can be analyzed by both GCMS in selected ion mode or HPLC by fluorescence without the need for a solvent exchange because the eluent is an acetonitrile. And the GCMS method is very rugged on a single quadrupole instrument. The extract is very clean and won't prematurely foul the GCMS system. Compared to other SPE methods, EasyPop NP gives better recoveries than silica gel SPE overall, and will give better recoveries for PAHs of 2 to 4 rings than MIP SPE. In comparison to silica gel and MIP SPE, EasyPop produces a cleaner extract and is easier and more versatile. There's less steps involved, less solvent usage than silica gel, and this produces an extract in acetonitrile which can be run by both GC or LC. So in closing, I thank you all for your kind attention. For more information on Supelco and EasyPop NP, solid phase extraction, or food safety, please visit the following websites. I hope you all have a great day and enjoy the rest of your August. Thank you. Now we will turn it back to Trevor.
Thank you very much, Jennifer. Just a reminder that the webinar video will be available early next week on Lab Manager's website. If you'd like a copy of the presenter's slides, we suggest that you reach out to them individually via email. Their contact information will be available at the end of the presentation. Our final speaker of the day joins us today from Metrohm. Dr. Stuart Procter graduated from Sheffield Hallam University with a degree in applied chemistry before going on to the University of Nottingham, where he earned his PhD. His research was multidisciplined, but the main objective was the sequence selective separations of oligonucleotides, After spending a year and a half in project management for a clinical research company, he joined the ion chromatography division of Metrohm UK. After enjoying 5 years of success as an IC specialist in England, he crossed the pond to throw his dice with Metrohm USA. As part of the field marketing team, he has helped with the development of new products and applications for new market initiatives. Thank you for joining us today, Stuart.
Well, thank you, and good afternoon, everyone. Thank you, Trevor, for that wonderful introduction. As Trevor says, my name is Stuart Procter, and I work with the Metrohm USA team. For those of you that aren't really aware of what Metrohm USA do, I have this opening slide to really show you the product ranges that we deal with. Typically, we deal with ion analysis, and going from left to right, on the titration side, we can deal with percent level ion analysis in multiple matrices, all the way through to the right-hand side of the screen, where we see the ion chromatograph and electrochemistry, which we normally associate with the sort of ppm and ppt level analysis. But you also have in the middle of the process monitoring for doing online level analysis. Now, of course, the focus of today is The focus of today is really talking about sample preparation techniques for chromatography. Now, Metrohm have a ion chromatography product range, and a lot of what I'm going to be talking about is how we can minimize lab time for the people in terms of preparation before the sample gets to the auto-sampler. So most of what we're talking about here is going to be inline or online preparation techniques to help minimize sample time and lab time for your technicians. The compact, so looking at our actual IC range, we start off with the Compact Flex. This is typically our unit which we'd use for a specific application in a QC type lab. We do, of course, have professional type research instruments which are very flexible. We can add conductivity detection for the ion analysis. We can add UV vis detection for chromophores. We can also put an electrochemical detector on there, as well as hyphenating to IC and ICP-MS type systems. Again, one of the big market trends at the moment is looking towards online type analysis. This will, of course, give great real-time feedback for analyzing processes. Of course, if something's truly online, the way you handle the sample preparation is absolutely critical in order to keep the system running and functioning as you want it to. Okay, so professional sample preparation. So most of my slides where we're talking about the inline analysis for IC and HPLC type systems, the left-hand side of the screen represents the auto-sampler. The right-hand side of the screen represents a chromatography type system. So, of course, we have the mobile phase going to the analytical pump, to the injection valve, to the column. Now, in this particular example, we're showing a suppressor and a conductivity detector. But, of course, we could put a UV vis detector if we're looking at chromophores. We could put an electrochemical detector if we're looking at redox chemistry. Or we could place a mass spectrometer there if we're looking at hyphenation. So the part that we're really interested in is this center block here. How do we get our sample from the auto-sampler to the injection valve and onto our analytical instrumentation with the minimum effort and cost benefit and time in terms of lab staff and processing samples? So the first example, which I'm going to talk about, is an inline ultrafiltration technique. And anyone doing any chromatography technique is surely aware that one of the critical or most important parts of the system is the actual analytical column. Typically, the particle size in analytical columns goes anywhere from 2 or 3 microns up to 9 microns. And obviously, you want to prevent particles from your sample reaching that column because it's very easy to block the column, and there you have to buy a new one. So if we look at this center block now, which really is a unique methodology from Metrohm that enables us to analyze a high volume of samples with a single 46 millimeter filter disc. So traditionally, for sample filtration, people do this offline using lower filters and syringes. Of course, if you're looking at a lot of samples, you have to go for there's a lot of time to physically filter samples. And of course, there's also an expense associated with those filter discs. So the Metrohm system is unique that we can actually do a high throughput of samples with one filter. Now, how it actually works is animated on the right-hand side of this slide. So we have 2 flows. We have a high flow and a low flow with a typical ratio of around 4:1. Now, what makes this system unique is that you can see that the actual sample flow is flowing perpendicular to the filter disc, perpendicular to the flow across the filter disc. So all the heavy particulates do not kick the filter and are transported directly to waste. Only a relatively small amount of sample is actually poured across the filter, and then that is then delivered to the injection valve, which enables us to do the analysis. So here we can basically filter down to 0.2 microns over 100 samples using a single standard 4.6 millimeter filter disc. So it's very versatile. Because we have advanced liquid handling on the Metrohm systems, we can also reduce carryover to zero by introducing multiple flushings of the filter in between samples. Now, very similar and in the same family as the inline filtration is our inline compact dialysis methodology. So again, if we look at this middle section, it has a very similar look to it. Instead of having a filter cell, we have a dialysis cell. This is typical for samples where perhaps the protein loading or particulates, so things like fruit juices and milks is high and you have emulsions. So the dialysis membrane removes all the high amounts of emulsions and lipids and keeps the system clean. And essentially you inject just the ions of interest onto the IC or HPLC system. So just to describe what we have, on the left-hand side we have the auto sampler and we have a flow which goes to the the dialysis cell and then to waste. On the right-hand side, we have a pump and a transport solution, an acceptor solution, which we actually dialyze into. That, once we've performed the dialysis, is transported to the injection valve, where it's then injected onto the LC or IC system. Again, we have a little animation just showing this. So again, you can see we call this stop flow analysis. So on the sample side, we have a continuous flow of the sample passing the dialysis membrane. On the right-hand side of the cell, we have the acceptor solution. So typically it takes around 10 minutes for an equilibrium to be reached. After that 10-minute time frame, the acceptor solution is then transported to the injection valve where it's analyzed on the IC. So typically that 10-minute time frame is only lost at the front end of your analysis because we can actually do the dialysis of the second sample whilst the first sample is actually being analyzed. This is great technique, and basically you would normally do this sort of analysis. You'd have to, say for example, if you're looking at a protein matrix, you'd probably add acetic acid to perform precipitation. You'd then perform a centrifugation, and you'd then take the supernatant and filter that, and all of that can be taken into one step in an online or inline dialysis preparation module. Now we move on to ways of eliminating sample matrix. Again, if we just look at this simple setup, what we've actually added to the sample prep section is a reservoir and an injection valve with a loop size between, well, any loop size you want from 10 to 50 microliters. This basically, well, hundreds of microliters, depends on the sensitivity you require. So what we actually do here, we use this transport solution to transport a volume of liquid from this injection valve in the center to the injection valve on the right-hand side where that chromatography system is, and there we either place an ionic trap column or a reverse phase trap column. And if we're looking at removing organic matrices, of course we would use an ionic trap column, so the ions are trapped on the column and all the organic matrix is flushed directly to waste. This transport solution can be water, it can be IPA water. So again, if you're looking at very hydrophobic drugs or molecules that you would normally have to perform some kind of extraction, instead of performing an extraction, you can just eliminate the organic material by pre-concentrating and flushing the excess organic to waste. So we have a slight modification to this technique, and this is called Metrohm Inline Neutralization. And as you can see by this slide, it's actually very similar to the matrix elimination. We still have the injection valve and the transport solution, but what we've actually inserted is a neutralization module between the injection valve on the center segment and the pre-concentrator on the right-hand segment. So again, we use the auto sampler to load up the sample loop. We then flick the sample loop and transport that sample through the matrix elimination module to the pre-concentrator. So this can have several effects. So if you're looking at a high ionic matrix, so you have a lot of cation interference, you can remove cations with this neutralization module, or you can also reduce the pH. So if you're looking at highly caustic samples or pH 14, you can reduce them to a pH of 7, which is going to give you a nicer, cleaner chromatography. And also on top of that, you have the ability to move things like amines and anything that's cationic, basically. So very similar to the matrix elimination technology, except we add this neutralization module between the injection valve and the pre-concentrator. So another very, very common way of eliminating a matrix is to perform a dilution. If you have the sensitivity in the equipment that you're using to detect low levels, you can quite often just dilute a matrix away. Again, so we can perform an inline dilution. We have two ways of doing this. We can do it in an external pot. So if you look on the left-hand side here, this is actually showing the external pot version where we do the dilution in a separate pot on the side of the system, which is then subsequently flushed. Or we can actually do a dilution directly onto the auto sampler and take sample from one tube and dilute it into another. So of course, this is going to save a lot of time if you're performing a lot of dilutions. And again, the dilute and shoot methodology is a very common way to try and eliminate matrix effects. We also have the flexibility on this system to auto-calibrate. So again, you can basically put one standard on the auto sampler, and this will then perform the dilutions to make the rest of your standards. And it also has an intelligent capability. So if a particular sample that you inject falls outside a calibrated range, it will automatically calculate a dilution factor that's required to bring that analyte back within the calibrated range. So again, it's a very powerful, very flexible system. So now we move on to complete robotic automation. And this is really when we're looking at solid samples, of course, which we can't inject directly onto an LC or IC system. Again, these configurations are incredibly flexible. If you look at that central unit, which is the main processing unit, that sample rack has filters on it, it has vials on it, it has beakers, it's got syringe filters. So again, these racks are customizable, so we basically tailor the methodology and the instrumentation to a particular sample for a particular analysis. So again, they're fully customizable depending on what you want to achieve. So the idea here, of course, is that you literally just put something like a solid or even a liquid directly into a tube, and all of the sample prep is taken care of by the system. So things we can do. Homogenization is very important, of course. If you're looking at tablets in the pharmaceutical industry, that first sample prep step is critical to making sure that we get a good mixture of your solution before we inject it. We can add solvents using our Dacino liquid handling technology. Stirring is also critical, of course, and that also goes with homogenizations. Of course, if you don't need a homogenizer, a simple stirrer may do the job. Diluting, again, is absolutely critical. We can change the volumes and liquids that we use here to get the sample concentration into the preferred range for the analytical instrumentation we're using. And pipetting also means we have liquid handling capabilities. So, for example, if you wanted to add a reagent to do a chemical reaction before the analysis to some kind of derivatization, or if you were looking at like a protein matrix, you could add some acetic acid and cause a precipitation of the proteins before you take them to the system. And again, filtering. Now, again, if we look at the slides, all the previous slides we talked about, all of those methodologies could be combined with this robotic sample processor, but we have filtering there, but that means we could also do things like matrix elimination. So just taking a quick closer look at a little bit of how it works. So you can see here we have a picture of the homogenizer. Again, it's a 25,000 RPM homogenizer unit, so it really does the job very quickly in a few minutes. There's also, if you look slightly to the left of the robotic arm, there's a rinse station, and again, that's critical to make sure that you have jet washing of that homogenization, the homogenizing unit in between samples, so you obviously don't want to get sample carryover. So once we've homogenized our sample, how do we get it to our system? There are several things we may want to do. The second tower on the left-hand side of the system has a lower fitting connector. So with that lower fitting connector, we can add syringes, needles, whatever you need. So typically what you do, you perform your homogenization, you'd add your solvents, you would then pull a certain amount of liquid into a buffer coil. You could then attach a filter to that lower fitting. And again, if you look to the left of that yellow bucket type thing at the bottom of the picture, there's actually a port there which you can actually transport sample then directly through the filter and to a HPLC system or an IC system for analysis. If you wanted to then not have the complete online solution, you could actually then pick up a needle and deliver sample into a HPLC type vial. So again, most people have standard XYZ auto samplers, and these are the typical HPLC vials that you would fit in them. So you literally inject the sample into the vial. The vials are then taken off the processor unit and placed on your HPLC system and injected directly. So to summarize, we've covered I know it was only a quick presentation, but we've gone through ways of inline sample preparation for simplifying the preparation from your auto sampler to your analytical instrument. And then we've gone one step further with a fully robotic sample processor, which can take solids, liquids, and deliver them directly to an analytical instrument. The overall goal here, of course, is to minimize, obviously, automation can improve precision and minimize the lab use time of staff in the lab. So just to finalize, or the last slide, Metrohm, start reaction. You can see the contact details for the company on the bottom left-hand side and my own personal contact details. Please feel free to reach out to me directly on my cell phone. Or, of course, you can use my email contact, and I'll be more than happy to help you if you have any questions. Back over to you, Trevor.
Well, thank you very much, Stuart, and thank you, Joni and Jennifer, for your presentations as well. If we're all ready, I think we'll move directly into our question and answer session, as we have a number of questions that have come in from the audience. I think the first question we'll start with is, how would you pre-treat a sample that's not in aqueous form to make it aqueous? Jennifer, could I start you off with that one?
What? I'm sorry. Did someone just jump in? I think it was Joni.
Oh, Joni, did you want to weigh in? Go right ahead.
I can answer that. In regards to SLE, how you would do that is through dilution. So you can go ahead, and if it's high organic, you might want to evaporate it down, reduce that. But if you bring it up by adding increases of water, which could be either acidified or basified, that will actually make it extremely aqueous. It doesn't have to be 100% aqueous. We've seen with SLE that you can use a 10% methanol or so, but you want to definitely increase the amount of water in the sample itself.
Great, thank you. Jennifer, anything, or Stuart to add to that?
Sure, absolutely. So I didn't actually show the slides for this today, but we actually have an automated extraction system. So basically you dose in your sample, assuming it's a liquid. You add a known amount of water. You actually mix the water automatically and then literally draw the supernatant, and you can then inject that directly onto an IC or an LC system. If you wanted to give further protection from the organic in the system, you could actually put that through the matrix elimination. So you would literally take the sample, mix it, you automatically mix it, and extract. Yeah, okay, but if you're looking at ions, that's fine. If you want to look at organics, then again, you could go for the direct matrix elimination approach and try and get it into some solvent and then inject it across a pre-concentrator or an ion trap or a reverse phase trap. So again, it depends exactly on the analyte you want to analyze. The extraction methodology works really well for ions. Obviously, if you're looking at more lipophilic and hydrophobic compounds, then one of the matrix elimination techniques can sometimes be utilized.
Okay, thank you. Jennifer, anything to add to that?
I was just going to say, well, a lot of it, with SPE, one of the main points is solvent exchange. So there's a number of different SPE techniques. So whatever your sample is in, you can do some sort of bind and elute strategy depending. I'm just talking very general here on SPE. You can bind the analyte to the sorbent and then elute it with a different solvent just to do some sort of solvent exchange. So that's it.
Great, thank you. Now everybody's, I think, touched on this a little bit, but how important is sample prep or sample cleanup for prolonging your instrument life or your column life?
Well, I noticed that. Oh, sorry, I didn't mean to interrupt.
That's okay.
I noticed that one of the first slides that Joni brought up was looking at an avocado extract injected onto a chromatographic system. And I know when we were looking at cleaning up avocados by catchers here at Supelco, we wanted that chromatogram to compare to the cleanup, and it was so bad that it actually shut down the system when they did the extract. They injected the avocado extract onto the GCMS system. It shut down the whole system, so we never got that chromatogram for comparison's sake. So, yeah, it causes a pretty big mess. And if you think about it this way, so if you have a really expensive car, say you have a Lamborghini, are you going to get in the Lamborghini with muddy shoes? No. You are going to make sure, to keep it pristine, and you don't want to mess it up. You're using these very expensive instruments like LC-MS, GC-MS. You don't want to inject junk onto there and get everything all fouled up when you're using something that's very precious. And I know many people, they have to jump through hoops for capital expenditures at their companies. So, it's better to do some sort of cleanup prior to analysis.
Thank you, Joni.
This is Joni, and I totally agree with Jen. And if I could just expand on that a little bit, it doesn't even have to be a difficult matrix like avocado. It can be something as simple as urine or plasma that many people will just dilute and shoot, and actually that will be fine at first. But over time, and this is what people really need to realize, the avocado example is a good one because you're going to see the problem right away. However, what we really see a lot of times out in the labs is that you'll do a dilute and shoot, and then over time you're starting to see chromatographic issues, and it will be slow. And so if you have very, very long runs, it might accumulate over time and cause a problem. So whether it's, the simpler matrices like urine or plasma or the very complex ones like food, it's really important to do your due diligence and do some type of sample prep to be aware and keep your system as clean as you can so that your data is worthwhile, then you don't have to repeat.
Great, thank you. Stuart, anything to add to that?
I think I'm on the same boat as these two. Essentially, any sample preparation that can keep the system clean from potential contaminants is always a good thing. I mean, obviously, as they say, things may look okay initially, but as time goes on, the evidence is remarkable, and you can save a lot of money in analytical columns, detectors, and that's really the whole point of doing sample preparation, is that you're trying to protect the analytical system from the nasty stuff you actually want to analyze. So absolutely.
Great, thank you. Now, are there any opportunities to perform some of the sample prep right at the time of collection?
I think it really depends on maybe the type of sample you're collecting. The only issue I have with that is many times you have to differentiate sample collection from sample prep. So, for instance, if we keep on the lines of the avocado, you're not going to collect one avocado. You're going to collect several kilos of avocado, and that has to be transported. With the issue of the water, there actually are some materials that are available that will actually collect a water specimen through a lake or through a river, and then that piece of material can actually, which is doing a type of sample prep or sample collection, and can be taken to the lab. But in general, unless you guys, Stuart and Jen, have an idea, usually the sample collection is somewhat different than the sample prep unless you're going to bring the instrument of analysis into the field with you.
I actually have a pretty good example. There's something called SPME, which stands for solid phase microextraction. And what this is there's a thin fiber that is coated with different sorbents, And you can actually take this into the field, and you collect the analyte of interest is concentrated onto the fiber, and then later on it's desorbed. So that's one way that sample collection and sample preparation can be combined in the field.
Great, thank you. Stuart, anything to add?
Not really on that one. I have to say, I mean, most of what we deal with is actually samples that arrive in the laboratory and how we take that from point A to point B. Of course, there are things we recommend to be done to samples to preserve samples before we analyze them. And quite often that procedure can actually cause more of a headache because sometimes we have to get rid of that matrix after we put it in there to prevent things from happening. So typically speaking, on the chromatography side, we don't offer much in that product line. But of course, preserving samples is one of the critical. You want to make sure that what you're analyzing is representative of what you actually took in the first place, and that's it's like preservation versus sample preparation, so to speak. So again, quite often the preservation can be more problematic for the actual analysis itself. So it's whichever way you want to look at it. But
great, thank you. Joni, you mentioned that catchers are expanding beyond the food industry. Where do you see the emerging applications for catchers?
We're seeing some in soil. We're also seeing it in the forensic type market. Not so much clinical, but more biological, where, again, going along the lines of I don't want to just do a dilute and shoot, but I want to do some type of, as I like to call it, a rough and ready cleanup. And so we're starting to see it in that area too.
Great, thank you. Jennifer, anything to add to that, or Stuart?
Catchers in general, we kind of think of it as enough sample cleanup. Like, it's not perfect, but it's going to, get you through. So I know one area in which catchers has expanded to. Generally, a few years ago, catchers was just viewed as sample prep for pesticide residue analysis in food. However, because of some new sorbents that have come onto the market, people have been analyzing like PAHs in hamburger or PCBs in milk. It's definitely expanding into the other areas. I know Joni mentioned many others as well, but science tends to expand its way into other areas, useful science. So.
Yeah, good point.
Great, thank you. Many of the methods you're describing appear to use very small sample volumes. Is this sort of a current trend for sample prep, and is that affecting the approaches that are taken?
This is Joni. I think, and this is just my response to that is, if you can work with smaller samples, smaller volumes, it's easier. That actually lends itself very nicely to automation, which takes a lot of the variability out of the manual workings. However, in fact, this is a really good question because I was just at IUPAC last week, and this was a big discussion as to how small of a sample can you take from, say, for instance, a crop or a fruit or vegetable and still have a representative sample. In general, yes, of course we would like to work at smaller sample sizes. Why not? It's easier to handle. The logistics is minimized. The cost is minimized. Automation is available. Less solvent usage. So all in all, it's definitely a very appealing approach. And I think in time, that is the direction that everything will go. And also, too, we've got the sensitivity and selectivity with these really high, powerful instrumentations. So it's all kind of moving in that direction. We're not there yet, but...
Yeah, I mean, I'd like to add to that. I mean, basically, the market trend is, as instruments become more sensitive and more powerful, we can introduce less and less sample and still achieve the results we want. Of course, if you're injecting smaller amounts, then there's less matrix to actually deal with in the actual preparation and purification step. So all of these methodologies we talk about have a capacity on what they can actually remove. So obviously a smaller sample means you can get more samples per, well, in terms of capacity in preparing the sample for injection. So yeah, it's always a case of, as I think it was Jody just said, that you've got to worry about the representative sample of what you're sampling as well, of course. So how do you get your sample representative when you're only taking 20 milligrams of something that's 2 kilograms? And it comes down to how homogeneous is the analyte of interest in the sample you're analyzing. So again, it's like a swings and roundabouts. Of course, smaller sample size is beneficial for the actual sample prep side because it's easy to prepare and much easier to get sample into the system. We can let the sensitivity of our equipment deal with the small quantities, but on the flip side, you do have to worry about getting a representative analysis of what you're trying to analyze.
Another issue with large samples is safety. For example, do you remember way back when large glassware was used to do sample prep? Some people had to climb ladders to get up there.
Yep, exactly.
The smaller the sample, you know, the easier it is to do that, as well as it's safer. There's fewer safety concerns as well.
Yep, exactly.
Great, thank you very much. When we're looking at automated sample prep, are we seeing more inline, just-in-time sample prep, or offline workstations and batch processing more valuable for some labs?
I think the general trend, this is Stuart, of course, the general trend in the industry right now is to try and minimize lab staff because of the overheads. And I think the more that's being forced into labs, the more the automation and automated sample prep and inline sample prep is growing. People are always looking at ways of maximizing the throughput. And of course, sometimes it's a case of trying to eliminate or improve the precision of methodology. So I think inline sample prep is certainly becoming very popular, certainly in high throughput quality control laboratories. Of course, in a research environment, it's a different kettle of fish, but it depends on the lab in question and, of course, the company that's running the lab. But certainly there is a strong lean towards improving ways of doing sample prep to simplify the analysis and also, obviously, minimize labor in the lab doing sample prep.
Yeah, I agree with that. We're seeing a trend that mostly pharma and clinical forensics labs, they tend to be moving towards high throughput type sample prep. For example, 96 well plates that can be automated and inline sample prep, inline SPE. So yeah, it depends on the lab, probably not so much in academia, but more so in the pharma industry as well as high throughput places.
Yeah, and this is Joni, and I agree with both Stuart and Jen. And then there's also another aspect is that, yeah, sample prep is or has been considered a bottleneck, but you also have to realize the reams and reams of data that these high powerful triple quads and ion traps and TOFs are producing, and that has to go through. People have to look at it. They have to evaluate it. They have to make sense of it. So if you can minimize the amount of labor associated with the sample prep, you can then have people looking at the data and getting that through and back to the customers.
Great, thank you. Now I have a question. Are there particular types of syringes that you recommend using for sample prep?
I think it would depend on what you're doing exactly. It's kind of hard to generalize. Stuart or Joni, do you have any suggestions?
I'm not sure what that question is referring to. Is that referring to a sampling, or is that referring to on an instrument for injection into? I'm sorry, could maybe they...
Maybe we need a little more clarification on that. You know, I will forward that. I will forward to that to you folks to respond to them and maybe get a little more information.
The Metrohm opinion is the Metrohm opinion on that is obviously the no syringe option is obviously the best option. So you know, if you can automate the sample prep to get rid of syringes altogether, then that's great. And that's really what we focus on when we're looking at our methods of improving analysis. Removing the syringe and filter is one of our key areas of interest. So, but yeah, I don't know. I couldn't add anything more on what defines a good syringe, so to speak.
Great, thank you very much. I think I have time for one more question here. There's a lot of talk about green chemistry right now, and there might even be some evidence that it's catching on in some places. Is this movement affecting sample prep? Are we seeing less solvent usage and that sort of thing going forward?
Most definitely.
Go ahead, Joni. Sorry.
Oh, okay. All right. Yeah, I'm sorry. Yes, exactly. I mean, one of the things, I'll just pick a couple of the techniques that I talked about from liquid, which was used huge amounts of solvents, and some of them extremely dangerous to be using as the amounts that they were doing, and also the cost of getting rid of it. And now you see people doing SLE, so that minimizes the solvent usage. And you also see catchers. The initial Luke method or modifications of it used huge amounts of solvents, and so the catchers was also a decrease. Also with SPE, going from larger cartridge sizes down to the 96-well plates, that works out all in that perspective. And yes, of course, that's exactly, and what Jen said about huge glassware, that's also what you're seeing. You're seeing more where everything's in a smaller size, it's much greener, it's less hazardous for those that are working with things, and so, oh yeah, most definitely.
Great, thank you. Any other additions there?
I just wanted to add, we have a number of requests from our customers regarding different procedures, you know, using methylene chloride or hot toluene. There's always requests for us to find different ways to do the sample prep methods with greener solvents, such as, you know, water, acetonitrile, or, you know, various other solvents that aren't as scary, I guess.
Stuart, any last words?
No, I think it's a general trend, as they both said. Essentially, solvents in the lab is something that people are trying to move away from. And, you know, obviously, when you're looking at any chemistry, the environmental impact is always going to be important because we need to look after the planet. So I agree. It's a slow movement, and I think it's something we should all encourage, of course, because I think ultimately it's very important that we can greenify ourselves, shall we say. I think I just made that word up, but I'm going to stick with it.
Good word.
It's a good one, Stuart. I like it.
Great.
Blended.
Well, thank you very much for joining us, and this brings us to the end of today's webinar. If you have any further questions, please consider reaching out to the panelists directly, as they are the true experts in the field. Their contact information, as you can see, is available on the screen now. If we didn't get to your question, I'm going to send them to our speakers directly, and they'll get back to you. Just a reminder that today's webinar will be available on the link you see on the screen. And on behalf of Lab Manager, I'd like to thank our panelists today for all their hard work. I'd also like to thank our sponsors, Agilent Technologies, Supelco, Sigma-Aldrich, and Metrohm for supporting our Tech Trends webinar series. As well, I'd like to thank all of you for taking time out of your busy schedules to join us today. You may wish to join us next month when our next webinar will focus on automated liquid handling. It'll be taking place Thursday, September 25th from noon until 1:30 p.m. Eastern Daylight Time. And for more information, you can visit our website at www.labmanager.com. Hope you can join us again next time. Thank you and have a great day.